Diagnosing, predicting, and tracing a disease are important aspects for its prevention and treatment. This is especially important for cancer diseases, as they are currently the leading cause of death in developed countries. How is this diagnosing, predicting, and tracing of disease possible? Measurements of either internal or external biological parameters can help the clinicians monitor and predict the health state of an individual or group. Any biological parameter which can successfully aid in predicting and monitoring of the health state can be called a biomarker. Therefore, biomarkers can be used to diagnose, predict, and trace diseases in general. More specifically, can be used for cancer prevention. An example of an internal biomarker is glucose in the blood. On the other hand, an external biomarker is the body mass index. Another important internal biomarker is the prostate-specific antigen, which is a famous biomarker used for prostate cancer screening. Being very widespread in clinics, biomarkers are very important for diagnostic and prognostic purposes. This begs the question, how are they discovered and routinely used? What are the necessary steps to develop an efficient and meaningful biomarker? In this video, we will explain how researchers develop biomarkers, giving successful and failing examples. The phases explained in the following part serve as a guideline for researchers when developing, evaluating, and implementing biomarkers. The first phase attempts to find potential molecules or other biological indicators. This is done through a lot of exploratory studies and experiments using various different methods and approaches. When studying diseases, scientists analyze DNA or RNA molecules, proteins, anthropometric data, or create images of the tissue or region of the body of interest. When searching for potential candidates, it is important to start from the biological function of the biomarker, whose measurement will then be directly or indirectly taken. In this phase, the researchers compare the direct measurements, for example abundance of a gene in tissue, in healthy patients against those that have the disease. The next phase measures the biomarker using available and non-invasive methods. Measurements can be either directly or indirectly taken. Direct measurements refer to those measurements taken directly from the place where the biomarker acts or functions. However, these measurements are mostly invasive, such as performing a tissue biopsy. Therefore, no-invasive or indirect measurements are preferred. This can be achieved by measuring the abundance of a biomarker in the urine, blood, or saliva instead of in a patient's tissue. In this phase, the researchers evaluate the efficiency of the biomarker measurement in both healthy and diseased patients. In the third phase, another factor is added: time. Researchers perform the biomarker measurement and follow the volunteers up until a certain point. Afterwards, a retrospective study of the following individuals is performed. The researchers check if the biomarkers have the ability to predict or diagnose a disease looking back in time. They want to know how far back a biomarker can be used before a disease develops. For example, in this part, the researcher would check if a biomarker was predictive of cancer development 2 years before its occurrence. The fourth phase attempts to refine and better understand the biological nature of the biomarker. This means that the scientists try to understand which subtypes of the disease are captured by the biomarker. The results and analyses are continuously being updated in this phase and enrolled patients are being followed. Finally, researchers summarize and evaluate the improvement in life quality gained by the biomarker, as well as its financial viability. This phase is continuously evaluated after biomarkers are available for the population. A good example of diagnostic biomarker that changed how one understands breast cancer is HER2, known as HER2. HER2 is a cell surface receptor protein expressed in several tissues of the body. A subset of breast cancer patients overexpress these proteins on the surface of the tumorigenic cells, and they have a poor prognosis. Knowing the expression of this molecule led to the development of drugs targeting this receptor. Nowadays, breast cancer patients are routinely tested for HER2 overexpression, and if that's the case, they receive a special medical treatment. This biomarker went through several of the described phases, the exploratory phase followed by the fourth and fifth phases. On the other hand, the advanced ovarian cancer marker CA125, a protein found in blood, has been proven to not be so useful as a screening tool. A recent study has shown that in a randomized clinical trial setting, screening for CA125 and second-line transvaginal sonography do not show reduction in disease-specific deaths between the screen and control groups. This is a biomarker that went through all the phases, and unfortunately it is not recommended to be used anymore. This shows the importance of constantly evaluating biomarkers. In this video, we cover the reasons why biomarkers are developed and why they are so important in the clinics. Their detection is a long and often difficult process that requires a lot of resources. In order to better diagnose, predict, and prevent diseases, especially cancer, the government, policymakers, general public, and researchers have to understand the importance of biomarkers as well as their development process.